have Professor Don Katz. Don is better, no? Than Donald, I guess.
Don was great.
Don Katz. So, Professor Don Katz, he is a professor of psychology at Brandeis University. He's also affiliated faculty at the Benjamin Mai Volunteer National Center Complex Systems, and also affiliated faculty of interdepartmental program in neuroscience at Brandeis University. He also had, he earned several awards, and I just want to focus on a couple of them. One is the Pollack Young Investigator Award for the Association for Perception Science. And I think what's great for us is a great lecture, and, and he was awarded by Janet Lerman, Newber 69, and Joseph Newber Prize for Excellence in Teaching and Mentoring. So it's great that we have him giving us the lecture. His main research is in the neuroscientific base for the taste and gustatory system. And he wants to understand what makes food taste good or bad. Mm-hmm. So please, the floor is yours.
Good morning. So this is going to be a little different. There's going to be very little math in what I present to you, at least on the surface. And I'm going to give you a view in on what, for this crowd, would be considered some fairly standard neuroscience. Although when I go and I present to the standard neuroscientists, they say, who is this freak and what is he talking about? And so I'm going to open up what I'm going to try to talk to you about today. My plan is to talk to you about some assumptions that underlie a lot of mainstream neuroscience work. And so I'm going to talk to you about some fairly mainstream neuroscience, and I'm going to talk to you about, I'm going to start with some potentially needless basics about the brain, because I understand that this is a group that is very diverse and mostly is not basic neuroscientists. That way, the way that most neuroscientists think, and I want you to know that what I'm going to tell you about is true even of a lot of neuroscientists who insist that it's not what they think, about modularity of function and responses of single neurons in the brain. And so I'm going to build up that story over the course of about an hour, I hope. And then I'm going to talk to you about some data, and of course I'm going to focus on data from my lab that challenges these assumptions, and that suggests that the things that the mainstream of neuroscientists believe probably are things that are incorrect and even holding us back. All right. And so to get started, and again, I apologize. Please feel free to let me know if I'm telling you stuff that is pointless. But neurons themselves, we're going to talk about parts, that the brain is made of parts. And to start with, neurons are made of parts. And so like there is the soma, the cell body of this particular neuron. And you can see that there's a lot more to this neuron. And you are probably all familiar with the fact that these neurons have, in addition to their cell bodies, they have a whole bunch of dendrites leading into the cell body and axons leading out. And then these neurons come in different types, such that while they all have the same parts, they themselves look different. And I meant to and forgot to get a picture to put in the middle here of what's called an interneuron. And so those are neurons that are found nestled amongst these types of neurons, but that have a very different shape and very different morphology and very different properties. And so neurons have parts, and then neurons themselves come in different types. So you can divide neurons into groups. And then, of course, and I should mention that these are images traced by Santiago Ramon y Cajal, one of the first great neuroscientists. And I find it interesting that he used this using tech from Emilio Golgi. These were two of the first great European neuroscientists, and just for a little background, they despised one another. And when Ramon y Cajal used Golgi's technique to make points, it made Golgi seethe. But so these neurons, though, it isn't just that neurons are made of parts. These neurons of different types, they have a way of communicating with one another. And again, we've been talking about this stuff already even in the previous talk, and I'm sure you guys have heard a lot of this. But just to make it clear, if you have a neuron, and this is my dopey illustration of a neuron, which is the dendritic tree and the soma and the axon, and so along that, you can see that you get communication of one neuron to the next at synapses, and so that means that within this one neuron, you get EPSPs and IPSPs. Those are excitatory postsynaptic potentials and inhibitory postsynaptic potentials, which are graded signals that lead to action potentials that travel down the axon. And so that is a pulse code, which is what we talk about a lot in neuroscience, which is the idea that these neurons communicate with one another by emitting essentially a digital response, which is a 1 or a 0 at each time point. The 1 is the firing of an action potential. And then at the synapse, there is synaptic communication, which leads to the next EPSPs or IPSPs. And so this is the basis of neural communication, and this is all voltage, and that's chemical. And again, I just wanted to make sure, I don't know where you guys are as a group in terms of all this, but when we talk about neural activity, we're talking about neurons communicating with one another via the integration of graded potentials that cause the output of action potentials, which then cause graded potentials in the next neuron over. And so that leads us to the basic analysis of neural activity, which is very far from what you just saw presented, where you are recording from a neuron and you're recording the pulse code. And so this is a single trial in which my postdoc put a drop of sugar water on an animal's tongue, and this neuron that he was recording from is in primary gustatory cortex, which is where I'm going to return to later in the talk. And so you can see the sequence of action potentials, and you can see, and that what you can't see because it's very variable, is that's when the sucrose was delivered. So there was spontaneous activity preceding, and spontaneous activity is just the word we use to mean we don't know what the fuck caused it. And so that is stuff going on that we're going to pretend is just noise, but probably isn't. But then in order to actually tell that there was a response here, because of that spontaneous activity, people typically will deliver many trials of a stimulus, do the same thing over and over and over, and now you can see when you have all these trials all lined up on when the sugar water is delivered to the tongue, you can now see there's an obvious response, and you can use what's called a peristimulus time histogram, which is peristimulus, meaning around the time of the stimulus delivery, and a histogram is just a graph of spikes per second. And so you can now summarize this firing. Whoop. Sorry about that. Something is going wrong with my presentation. Okay. And so you can basically summarize the response of the neuron that way. We can even just draw the envelope over it. And that envelope is basically a sugar response. It's a response to sugar on the tongue. And just to make sure everyone is on the same page and that you all know, that is an excitatory sugar response. It is absolutely possible to get an inhibitory response, where the firing rate decreases when the sugar was delivered, and that is still a sucrose response. And so most neuroscientists, when they are talking about responses, are starting with something like this, that there is a pileup of action potentials after a sensory stimulus was delivered, or after the animal, or when the animal was doing a particular thing. And those are what we talk about in terms of neurons responding. And so, of course, these neurons come in groups. Their somi congregate. Boop, 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 boop. There we go. And so this is another picture from Ramon y Cajal showing a bunch of neurons in the same cortical slice. And so when you have a group of somas, they will tend to appear in the same spot. And you can see, in fact, that there will be, now it doesn't mean that the neurons themselves are all there, because of course neurons spread out their dendrites and their axons, but we identify parts of the brain in terms of groups of neurons, somas, being around in the same spot. One thing you can see here, this is the cortex. You can even see that within this cortex, there are the stereotypical layers of cortex, and I used to have arguments with Asif about whether you should think of this as a single bunch of cell bodies, or whether you should think of each of these as separate structures. But the point is that when you get neurons together, they form into groups, and then those groups form into networks. And so this is a picture from Van Essen's drawing of the visual system of a primate. And he mostly did this just to shock you, just to say, oh, look at how big it is. But each one of these little boxes is a group of cell bodies that have congregated, and each one of these lines are a bunch of axons connecting two places where cell bodies have congregated. For reference, right here, that's primary visual cortex, which we're going to talk a little bit about as we move on. And so, again, brains, you can break things down into parts, and one of the ways people break things down into parts is that the congregations of these cell bodies gives you regions, gives you cortical regions, gives you subcortical nuclei. Basically, that people think of the brain as being a collection of different brain regions that are connected by axons. And so just because I'm going to talk a lot about taste, here's my own little dopey diagram of the taste system. One of the reasons I study taste is because it's a lot less complicated than studying vision, but there are other reasons as well. Lacking from this, which is going to be one of the main points of the talk, is that there's a lot of feedback in this diagram that I've left out. But again, people talk about the taste system in terms of different brain regions, which are collections of cell bodies connected by axons. And so the real problem with this is that once you've sort of accepted that you can break brains into parts, then it becomes natural to think you can break function into parts. And so if you have different brain regions and you have a complex behavior by an organism, or you have an organism doing something, why not take the function of what the animal is trying to accomplish, of what the organism is doing, and put different aspects of that into different places? And moving beyond from that, since there are different types of neurons, and neurons that look different and fire different and have different proteins within them, why not break function down and say this neuron type is doing one thing and this neuron type is doing another? And in fact, that's what the field does. And so I'd like to take a brief, almost philosophical foray into the fact that this sort of suggests you have to ask the question, how do these brain regions and these neurons work together? And when you say parts working together, it's a very simple thing to say, and we all think we know what it means. But here I'll just list a set of things you could mean when you say parts are working together that are very different. Parts could work together like parts of a car, each of which has a specific function, and the functions are used together. The carburetor and the engine do different things, such that if you were a very good mechanic, you could actually take the carburetor to one car and figure out how to put it in another car. It would do the same thing there. It has a discrete function. But not to take too much time on it, but you could also say that parts work together like words work together in a sentence, where each word has its own discrete meaning, but in fact, that meaning can be altered by the other words it's in a sentence with. If I say to you, my room has bugs in it, you might think, well, that means there are insects crawling around my room. But if I start with, I'm worried that I'm being watched, my room has bugs in it, then suddenly we're talking about a different meaning of the word bugs. Bugs no longer has the same meaning because the other words that it's surrounded by lead you to think that we're talking about microphones. Or parts could work together like voices in a chorus, where each voice in a chorus has a different sound, but you don't know what you're hearing unless you hear the whole chorus. If there are any musicians in the crowd, if you have somebody who's singing a concert A, you don't know what that's going to sound like until you know whether the person above that person is singing a C, which would make it basically a minor third, or a C sharp, which would make it a major third, because those two sound very different. They have different meanings to us. You can go on and say that pieces of the brain could work together like pieces of the jigsaw puzzle, each of which contributes, but none being particularly meaningful in itself. We could talk about parts of the brain working together like transistors in a computer, computing a mysterious bit to a task at hand without being very different from any of the other transistors. We could move up to, they work together like molecules in a gas, where the function of temperature only emerges from the presence of a bunch of them. So when we say that parts work together, we could mean a lot of different things. And in fact, one left out of this is I always like to, after this, when I do this sort of lecture, usually I then follow this with showing an Escher print. An MC Escher, you can look at any particular spot in an Escher print and think you know what it means, and then you back up and you discover that it doesn't mean that at all. And so all of this is just to sort of make it clear that where we're heading with this talk is to make fun of the status quo, because what you're going to find is that most neuroscientists really just go with option one. That because we can say there's a group of cell bodies over here, and there's a group of cell bodies over here, this one probably does one thing, and this one probably does another. And why do they do that? Well, they do that because it's easier to think about it that way. And we like simplifying our theoretical constructs, and we like simplifying what we're having to do because the brain is so complicated. And also because our basic techniques bias us in that direction. And so the basic techniques of neuroscientists are made, they don't have to do this. In fact, I'm going to show you examples where we do the opposite with it. But for instance, electrophysiology basically gives you data on what these neurons respond to. And so it's very easy. In fact, it's hard to resist when you record from one group of neurons over here and another group of neurons over here, that the take-home message is going to be what's the difference between what these neurons respond to. And therefore, you're going to say, these neurons are involved in this, and these neurons are involved in that. Similarly, neuroscientists use a lot of experiments that involve lesions, brain lesions or brain inactivations. And that is where you either destroy or put to sleep some spot in the brain. And then you say, what happens when you remove those neurons? And the answer is going to be either nothing or something. And if you get something, then you say, okay, these neurons do that. And if you get nothing, you say some other neurons do that. And left out of there is all sorts of things, like we actually have several studies. Once you start questioning this stuff, you find a lot of studies where removing a group of neurons actually, rather than doing nothing or fucking with the animal, they actually make the animal better. But so I'm going to just give some examples. And I could give thousands of examples, because as I say, this is where the field is. But I want to give you some examples just so you can see, and I'm going to give you examples that I like. Just a couple of examples that I like. And I like them because these are people who are not idiots. And these are people who didn't just do the simplest thing, and they actually worked very hard, and the results are great. But what the results are is straight out of this basic bag, where the whole point is figuring out what brain areas do what and what neurons do what. And so one example is from eye blink conditioning and the cerebellum, a field that I did my graduate work in. And so to acclimate you to this business of eye blink conditioning, let's say you are a bunny or a person and time is going on. You can measure your eyelid opening and closing. We do that in any number of ways. That's no problem. But so in this graph, as you go, this line goes up, the eye is closing. Up at the top is when the eye is shut. And so if you, for instance, were to deliver a puff of air at a person or a rabbit's eye, you would cause an eye blink. And now if you do that, but every time before you deliver that air puff, that's a naive blink, that all of you will do that. If before that air puff you play a tone, and by before, I mean a quarter of a second before. So this is something that actually my bunnies did better than humans, because humans would try to make it a cognitive task. But with a quarter of a second, it's not a cognitive task, or at least not in the classic way human researchers like to think. But if you do that for basically a couple of sessions worth, what ends up happening is the animal blinks to the tone. And the animal has learned a conditioned eye blink. And so this is a learned blink. And you'll notice that what happens is the eye closes when the air puff arrives. The animal has learned to avoid the air puff. And so this has been studied for years. Just as a side note, we used to despair for why we were studying conditioned eye blinks, which seems to have nothing to do with the real world at all. We used to make up stories about how maybe the bunny is like running through a forest, and when it hears a twig snap, that means it might get hit in the eye. In truth, it turns out that what probably is the important thing about this behavior is not tone-driven blinking at all, but the fact that if something suddenly looms at you, you will blink. And this is a behavior that is very important for human beings and other animals. And it turns out that brand new infants do not have this reflex. And so they have to, that's what they learn. And this is probably just us hijacking that to study something altogether removed from the real world. But this has been studied to death, and what has been shown is essentially that information about the air puff and the tone converge within the cerebellum, and in particular, most important for us, they converge within the deep nucleus of the cerebellum called the interpositus nucleus, which outputs to a brainstem motor relay that then connects with the eyelid musculature. And so while these blinks, the naive blinks, do not require the cerebellum, it turns out that the cerebellum is thought to be the place that takes care of the conditioned eye blink. And the experiment that did that is the experiment that's most well known for that, the most famous one, work done by a friend of mine named Dave Krupa. And it's just a lovely, elegant experiment using lesions. And so what he did was he set up groups of animals in which he infused mucimol, which is a GABA agonist. And now we're going, I don't know if any of that means, what that means to people, but for our intents and purposes, a GABA agonist causes activation of all the inhibitory synapses on any neuron, which all neurons have, and basically hyperpolarizes the neurons and makes them unable to fire. So it is a functional lesion that lasts for a few hours. And so we had some animals that were intact, and you can see that those animals, across training sessions, learned to make conditioned eye blinks at a very high rate, and by high rate we're talking here 90% of the trials. And then there were some animals in which he knocked out the interpositus nucleus, and across the 6 training sessions, those animals learned nothing. Those animals did not become able to blink. And then in a third group, because he isn't an idiot, he also took and knocked out the red nucleus and showed that those animals also learned nothing. But then what makes this study really elegant and good is that then he stopped mucimoling these animals, stopped infusing mucimol, and what you can see after that, and so again, this is the interpositus data, and that's the red nucleus data, but what happens when the animals are now intact? Well, for starters, the animals that had learned have still learned. The cool thing is that the animals that had red nucleus knocked out didn't need to learn. They basically immediately hopped up, and their performance was at max. So as soon as they let the red nucleus go, these animals were actually turned out to be conditioned, and the conclusion was that the red nucleus isn't responsible for learning. In fact, all it's responsible for is communicating what's learned above out to the eyelid. And so the animals had learned, and all you had to do was let that pathway free so that the animals could show it. And in the meantime, with the animals that had the interpositus knocked out, what you can see is that after the mucimol was removed, now they learned in a function that looked almost identical to naive animals. And so basically, this is a very elegant study using functional lesions to basically make the point that this is where learning occurs. And that's where neuroscience is. I'm going to give you one other I'm going to skip, actually, the electrophysiology data and move on to one other example, which is another favorite of mine because it connects up with my work, which is the neural control of emotion. And this is also very elegant old work where a researcher named Antonio Damasio in Iowa was trying to figure out how do you actually study emotions in humans, which is a fiendishly difficult thing to do because no one will tell you the truth about how they feel. And so he developed an operationalization where he basically gave subjects a game to play that involved 2 kinds of decks. This is called the Iowa card sorting task. And there was one deck that when you pick it, it would tell you whether you won or lost money, and it would give you big gains, but then much bigger occasional losses. And so if you chose from that deck, you'd eventually be an unhappy person losing money. And the other deck was much less fun. It had very small amounts, but if you kept going from that deck, eventually you would end up in the black. And of course, one of the most amazing things about this is of human subjects in a situation where they're not using real money, but the amazing thing is that people got really, really invested in this game. And what you find is that most people learn over time to go for deck 2, but it turns out that so what happens, in fact, is you have deck 1, and if you choose from deck 1, then you have a really unpleasant experience when you've lost money, and that is an emotional response. And over time, that emotional response becomes an emotional response that you experience even before you choose from the deck. And so people would report that even hovering their hand over deck 1 made them feel bad, and that was eventually what led them to choose from deck 2. And the reason I'm telling you about that is that patients with cortical lesions, in particular this part of cortex was a prefrontal cortex, basically never learned. They consistently for deck 1, so something about having a prefrontal cortical lesion makes you unable to do this task. And it's not a memory problem because these people could verbalize the outcome. They could say, okay, I'm losing. This is a losing deck, and it just didn't help them. They also, by the way, could do other types of memory tasks. And so one of the things that makes this experiment elegant is that Damasio went to an entirely nonverbal measure of emotion in order to figure out what was really going on. And this measure of emotion is called galvanic skin response. And basically, you can put 2 electrodes across your skin, and you actually have a conductance of your skin, and that conductance changes when you have a strong emotional reaction. If I was to do to you guys what I do to my undergraduates and suddenly scream, you would find that you experience a galvanic skin response that you can even feel. I'm not going to do that to you. But this sort of thing gives you a hint that the subject is experiencing a strong emotion. And so there's been lots of studies done on this. Like when you speak a neutral word to someone, you get a small galvanic skin response change even to that. If you were to say to them some very, very dirty word, then they have a strong galvanic skin response. And hovering in the middle is if you use a euphemism. So this would be the word fuck. This would be the word copulate. And so galvanic skin response is a good way. It's not a good way to tell you what emotion the person is experiencing, but it lets you know that the person is experiencing an emotional response. And by doing this, they learned something about what's wrong with people with prefrontal cortical lesions. And so following a big loss, they pick from deck 1 and they lose money. Control subjects, normal people, show a strong GSR. They show a strong galvanic skin response. It's a negative experience. And people with PFC lesions also show a strong galvanic skin response, so they actually do experience that as nasty. And just as an extra control group, they also ran a bunch of risk takers, people who like doing frightening things, like taking risks, and they also show a strong galvanic skin response. So these people aren't risk takers, are still experiencing the emotion, they just like it. But it turns out that when you look at whether or not preparatory to that deck choice, you have that emotional reaction, control subjects show that strong GSR. So the control subjects do exactly what I described there. When they're hovering over that deck, their galvanic skin response shows that they are having an emotional reaction to that, and that's what eventually pulls them away from it. And that's where the people with cortical lesions have a problem. They show virtually no GSR. And by the way, risk takers show a strong GSR. So Antonio Damasio concludes that PFC patients experience emotional valence but can't use it to drive behavior. And so, again, what's going on here is standard neuroscience. He's trying to break down what about the PFC, what is the PFC doing in this task? And it turns out it's not as simple as they can't experience emotion. They can't use the emotion that they experience to drive their behavior. This becomes important because it turns out that amygdala lesions and cortical lesions cause similar performance deficits. People with amygdala lesions, and amygdala is the part of the forebrain, deep in the forebrain, part of the limbic system, that has often been associated with emotions. And it turns out with people with amygdala lesions also don't learn the task, but they're slightly different. When you talk about preparatory to deck choice, which normally causes a GSR, people with amygdala lesions, unlike people with, like people with PFC lesions, show no GSR. But when you talk about following the choice of the big loss, remember that the PFC lesions really don't impair the GSR much, but in amygdala patients it does. And so what Damasio concludes is that the amygdala is involved in setting emotional valence. That's what the amygdala does. And that information is used by cortex to drive action. And so what we've done with these natural lesions and some clever experimentation is we've said, okay, in terms of emotion, we can now tell you the separate things that the amygdala does and that the cortex does. And this is the way neuroscience has gone. And again, this is not a course about emotion. This is not even a course about neuroscience. I'm just setting up for you why it actually, is reasonable to think, excusable for people in neuroscience to think this way. And every now and then, there we go. So this idea, though, that you can break up the brain into brain regions and use those brain regions to functionally come up with function, basically the logical idea that if you've got a task that you can divide into aspects and a brain that you can divide into parts, it's natural to make connections between those. That has begat basically a reductionist trend to try to say, to go down to each neuron type within a brain region has a separate function. Because like the brain regions can be separated, neurons can be separated into separate groups, and so why not do the same thing? It's also worth noting that the hippest modern tools of neuroscience further encourage bias in this way of thinking. And so as an example, we are in the midst of the molecular revolution. We can now, in neuroscience, make use of the genetics of an animal and of the makeup of the neurons to grab a hold of individual neuron types, to say we are just going to look at pyramidal cells in the cortex. We're just going to look at this type of interneuron, and we're going to manipulate that type of neuron. So essentially, we are now capable of doing lesioning and inactivation of individual neuron types, thanks to the molecular revolution in neuroscience, and that basically gears us to make distinctions between neuron types by the same logic. And so all you need is knocking out one neuron type to have a different impact than knocking out another neuron type, and you can then make a functional distinction about what each neuron type is doing. Also, because a Katz talk wouldn't be complete unless I bash on calcium imaging. Calcium imaging, which is the current favorite technique for looking at what neurons do, is also a force in this direction, because modularity quietly assumes the idea that you can break function down into this area is doing this by itself and this area is doing this by itself. It quietly assumes a relative lack of interaction dynamics, that these two brain areas aren't doing anything that needs them to communicate until they've both done their job. So basically, in the example of Damasio's work, the amygdala figures out what the emotion is, and then passes it to cortex, and cortex works with it. And never is there really an interaction between the two other than the passing on of information. And so with that sort of thing in mind, a brain area and a neuron either does something or it doesn't do it. And so calcium imaging, which has very low temporal resolution and low signal-noise ratio, means it's mostly good for concluding either these neurons were active or they weren't. And so calcium imaging, with which you can't even look at fine-grained interactions between neurons and between areas, reinforces the desire to say, hey, those neurons are doing that. Why? Because they respond. Essentially, you've reduced what you're saying about a neuron to simply, does it go on in this situation or does it not go on in this situation? Is it doing something or is it not doing something? And so I'm going to give you an admittedly extreme example. And this is from my field, and this is work from Charles Zucker and his group. And what it is imaging from primary gustatory cortex, primary taste cortex. And just to give you an idea of how calcium imaging changes the game, basically the information has been reduced to pseudo-coloring of the neuron cell bodies that were significantly active when sugar or a sweet was put on the tongue. And that is the entire data set. And so they are showing that there are a bunch of neurons that fired, and the claim is that very few neurons fired to other tastes. And so these are sweet neurons, and that's what they do. Yes?
Can you say a few words what you mean with this is the entire data set? Because clearly it isn't, right? There is dynamics, they just chose it in this way because they're
No, absolutely, and that's sort of my point, is that the reason I gave you all of this stuff that really is from very particular disciplines and isn't specifically what any of you are interested in, is to sort of set you in the mindset of the standard neuroscientist that we don't need to know that stuff to understand what's going on. And so this is the extreme conclusion where basically you don't need to know any of that. And what he can report is that he can find parts of the brain, parts of gustatory cortex that only respond to sweet stimuli, parts of gustatory cortex where neurons only respond, and again, responding, ignoring anything but does it go on or not. And then he can put together a map which says there's a part of this cortical region that does sweet, there's a part of this cortical region that does salty, et cetera, et cetera, et cetera. But lest you think that this extreme example is somehow bizarro, let's go just to standard classic studies in the visual system where they used electrophysiology, and so they were recording what was going on from this neuron from millisecond to millisecond, and nonetheless the data were reduced to simply how much does this neuron fire in V1 to different stimulus organ orientations. And so this conclusion is this neuron prefers this orientation. From there, it's easy to go to it is involved in coding that orientation. All that this cares about is a single number. How many spikes did this neuron fire in response to that? This is classic work. This is Nobel Prize-winning work where, again, the assumption is we don't need to know anything more than this. And it goes from the first cortical region to the last cortical region. This is data from Nancy Canwisher. Actually, this is not specifically Nancy Canwisher's work, but she is one of the biggest of the big shots in this field, looking at neurons in area IT, which is the end of the processing stream as it's described for vision. It is the fusiform face gyrus, and neurons in this region will respond to one particular face. And so this is a Steve Carroll neuron, a neuron that codes Steve Carroll's face. Don't worry, they also found Jennifer Aniston neurons. But again, while you can see that there is spiking patterns, the conclusion was simply, all you need to know is that this neuron responds to Steve Carroll and it doesn't respond to other things. And so the conclusion is it's a Steve Carroll neuron. So it isn't uncommon. And as a side note, I was talking to Asif earlier how I frequently find myself being on the outskirts of whatever community I'm lecturing to. And I was invited to a meeting in which there was lots of very nice modeling of visual processing going on, all of which started with the idea that the way to model visual V1 is in terms of neurons having just certain firing rates to each stimulus, and that's all you need to know. And so that's where the field is. And so to be honest, I have reached the end of the first half of my talk, and so I am happy to answer questions. If you have more questions, I'm happy to just keep going. Mm-hmm.
I want to push back a little bit on that this is where the field is, because there are so many amazing techniques, particularly in the context of calcium imaging or modern neuropixels, that have allowed for the first time probably in human history to record from thousands of neurons simultaneously in awake and behaving animals doing complex tasks. And we're developing the right tools at the moment to better understand the coordinated activity patterns. The reason why Hubel and Wiesel did this 70 years ago is because that was the limit of the technology of their day and age. They just couldn't do much beyond looking at single cells in anesthetized cats with carefully controlled visual stimuli. But it feels, at least to me, like the community in neuroscience is moving very, very rapidly towards these more advanced technologies with larger neuron numbers recorded at the same time.
So first, I will agree with the very first part of what you said, which is that neuroscience is a big field. And there are people doing all sorts of things, some people doing better work, some people doing worse work. But I will push back on the rest. These new technologies, what I'm pointing out is that these new technologies bias us further in the standard direction. And I invite you to go look at the journals that are considered the most important journals, your Science, your Nature Neuroscience, your Cell and Neuron, and you will find there those techniques that you describe, calcium imaging, neuropixel, used to reach conclusions about what this group of neurons does and what that group of neurons does, and it involves taking those data and reducing them down to, are they active or are they not? Only a few years ago, my department hired a new position, and we had half of our applicants, and this again was 3 or 4 years ago, were what we started referring to as the calcium imaging boys. And they had different systems, and they all were using optogenetics and calcium imaging and some great molecular techniques. And they were all reaching conclusions about, okay, it's these neurons that do that. And so the stuff that I'm laying out for you is stuff having to do with philosophy, with the way people look at them. It's not not only don't the techniques necessarily drive us into new realms, the techniques actually have been used to reinforce old thinking that was classic here. We have and again, going back to the beginning, yes, there are lots of people who are doing things different, but the mainstream in neuroscience isn't moving forward in the way you describe. And in fact, I've been to several meetings where they talk about all of the new stuff that's being done, and then they ask them, why is the progress not seeming to be, of breakthroughs not seeming to keep up with that? And the answer is because we're still working in the same regime. Question? Okay.
What you're bringing to us is something that's been present since the beginning of neuroscience. I mean, the idea of holism versus functionalism, and by the beginning of the last century, people trying to find the region for wearing hats in the brain or something like that. But why do you think that historically we've come to this idea? Rather, we've been through like Alexander Luria's idea about like modularity and complementarity, but we are still trying to find this like very specific regions rather than thinking about plasticity and multiplicity of utility of a single area. Why do you think historically we've come kind of backwards in this direction?
It's a great question. I can't pretend to have an answer. I think that you can look at societies in general and see that we keep returning to the same mistakes and the same bad ways of thinking. And again, in response to the earlier question, I don't want to suggest that there isn't a bunch of neuroscientists, even in this room, who are thinking differently from all of this. But whatever the reason, my argument is that the new stuff that's out there is only serving to, is being cycled back into these old ideas rather than driving new ideas. Having said that, the last half of my talk is about a bunch of data that isn't as theory-rich, isn't as mathematically rich as the stuff we've been talking about in here, but that does drive it in the same sort of direction.
I wonder if we can comment on this, just to, maybe as a response to Italo's question. Do we go down these different roads because we're limited by the metaphors we choose? So
I would say that that is absolutely a big reason. That we, in fact, we are still, I refer to these as the computer metaphor. We have computers and we love them, and now we have handheld computers and we love them even more, and we tend to use that metaphor inappropriately to describe the brain, which is where we're going to end up at the end of this, is that, but this is just coming back, this really just is another way of saying what you're saying, which is we just keep returning to the old ideas.
So my question is trying to understand the philosophy of your approach, because we can say, okay, there's the Steve Carell neuron, and there's the hat-wearing neuron, and each neuron or cortical region has a certain function. But a similar idea, which just takes it up to another level of abstraction, is saying that you have a network for which this eigenvector identifies Steve Carell, and this eigenvector identifies hat-wearing, or you can go to a higher level of abstraction, say I have this fitness landscape, and this local minima corresponds to Steve Carell, and this local minima corresponds to hat-wearing. So my question is, what you're trying to do here is to find this new level of abstraction where you can identify Steve Carell to some more abstract conjunction of neurons and regions, but still identifiable as a single thing? or you're going to an entirely different approach where there's no single identifiable structure associated with Steve Carell or Hatwari.
And so where I'm heading with this, although to be fair, I'm not going to return to the Steve Carell neurons myself, is sort of the latter idea that you came up with. What I want to suggest ultimately, and what I want to maybe have the last discussion is all of what's found. Actually, give me 1 second. So what we've done is we suggested that in the establishment field of neuroscience, there's a large percentage of that group that thinks that each part of the brain has its own function. Even neurosystems neuroscientists typically think this way. The new popular techniques reinforce that thinking and bring it down to the level of neuron types. The functioning of each neuron can be adequately described with a single number. The degree to which a stimulus turns it on. Again, the most popular techniques reinforce and further simplify that thinking. It all comes down to coding. It all comes down to the idea that the reason that you know that Steve Carell is there is because the Steve Carell neurons fire. And I don't care, and honestly, you can say, no, it's because the Steve Carell network fires. Fact is, you're saying that that what you're looking at is you're looking at the code that does the work of letting us know that Steve Carell is there. What I'm coming around to is the idea that we're better off thinking of this not as a code, but just as an effect of the system function. And so that trying to get away from thinking that these data, which are solid data, there really are neurons that respond only to Steve Carell, that they're not doing any work in the brain process that we're talking about. They are just an effect. They are exhaust from the car. Does that help? Does that answer your question at all?
Well, it starts to, but I guess I'll have to go through the entire lecture before we figure out how to do it.
All right, then why don't I keep going? All right, because now is for the happy part of my talk. And an embarrassing amount of neuroscience work is grounded in that computer metaphor. And so I am the only person that you will ever meet who is actually more negative than Asif and his students. But now I'm going to give the half of the talk that is about what I think is going on, that leads us in this new direction. And the only sad thing about all this is that with all of this stuff existing, the amazing thing is that it isn't having an impact on the mainstream of the... It isn't having the impact that you would like to think optimistically it should be having on the mainstream of the field. And so the importance of temporal coding sort of has been already, we've had people ask about the dynamics, lies in the fact that it challenges this status quo. And so there are many, many studies of temporal coding, although most of them, I would argue, miss the point. They are studies, analyses done by researchers who are still enmeshed in the idea that the neurons do something, that they're coding something, and the only advance is that they say that this representation unfolds over time. And I would argue that that's not only not, and while that sort of appealing, particularly if you study language and the idea that what you mean as you speak unfolds over time, it's actually untenable with regard to brain function. If you actually had to wait for a temporal code to evolve before you could know what stimulus is there, then you would probably not survive to pass your genes on to the next generation. But what is more telling is work showing that response dynamics are perceptually meaningful, that the dynamics actually are doing something, and showing that those response dynamics reflect the nature of the circuit. And so to start into that, I actually want to present some work that I have argued with Ghazenfar about, and I'm going to say up front that this work was done on extremely small amounts of data. And so I'm going to extrapolate the most positive and optimistic output interpretation of it that I can. But if you look at this study, you've got to be pretty forgiving of the fact that they're working with very little amounts of data when they reach the conclusions they were reaching. And so what this is work by a group, first author Sugasi, over 20 years ago, who were looking at the same face responses that we were talking before, only they were looking at them in macaques, and the macaques were looking at macaque faces. And what they found were face responses. But what they found was that, that the description of these things as just face responses misses something important, which is a meaningful dynamic flow as these faces are being processed. And to summarize, and again, with a very optimistic view, what they found is that as time goes on, and as time goes on, and again, one of the hard parts for a paper like this is that they're working with very small amounts of time. I'm going to go on to our work in the taste field pretty soon. I'll tell you, one of the reasons I went into taste is because I wanted to study dynamics, and I wanted chunky periods of stimulus response. And in the visual system, it all happens very fast. And so, but as that time progresses, if you look, or when they looked very carefully, what they saw was a sequence of responses, which is to say, if they looked at what the neurons were coding, that coding changed from moment to moment. That the first thing they saw was an initial response that essentially coded the fact that it was a face. So initially, for a moment, there was a fact that it was a face, and neurons would respond to almost any face briefly. And then, a couple, like 50 milliseconds in, that response changed to, hey, it's my buddy from the colony, Ralphie. And so suddenly it was a true taste response. Ralphie might be the Steve Carroll of the monkey colony, and this neuron is responding to Ralphie, but things didn't stop there. Then, 100 milliseconds later, the response has shifted again, so now it's only responding to Ralphie making an aggressive facial gesture. And so now it's an, oh my God, Ralphie's pissed response. And so what you see when you actually look at this more closely is that this is not coding a face. What it's doing is it's giving you an eye into the important process that a monkey undergoes while it is looking at a face. Because to be honest with you, it just isn't important in this monkey's world just to identify a face in terms of who the monkey is. The most important thing in the monkey's world is, am I about to get my ass kicked? Do I need to run away from this monkey? Do I need to mate with this monkey? And so what they were seeing in fusiform face gyrus was, and I'm not really arguing that it was a code for this, they were seeing a process occurring as the animal went from the input to the output, which is either running away or approaching. And so this brings me to my work, in which we see something very analogous, but we have much more data to work with, and so it's actually easier to see what's going on. What we do is we put electrodes into gustatory cortex. We put actually ensembles of electrodes into gustatory cortex. We don't use neuropixels for reasons that I would be happy to explain, because I'm going to have to on my next grant proposal, because where we've reached, the point we've reached in this faddish world of neuroscience is I'm going to have to defend my refusal to use neuropixels in my next grant proposal. But what we do is we record from squads of neurons, and if you just look at a single neuron in GC, you see something that's analogous to what I just told you about fusiform face gyrus. When we put tastes on the tongue, we see a sequence of things happening. And this is the recording of a single gustatory neuron. This happens to be the honest-to-God representative neuron. Most of the times when you hear people saying this is our representative neuron, they mean this is the best neuron we ever had. But what we see, what I've done is I've overlain a bunch of PSTHs, again, peristimulus time histograms, to a bunch of different tastes. And so this is this neuron's response to a bunch of trials of a bunch of tastes, each delivered individually. And the tastes run from sucrose, which is yummy, it's sugar, to quinine, which is entirely bitter and awful, although it does taste delicious with gin. And in between, we have a range of concentrations of sodium chloride, and we like these stimuli because at a very low concentration of sodium chloride is very palatable. In fact, this is normal saline. This is essentially the concentration of your bodily tissues, and you love normal saline. Even though you don't get a chance to drink it very often, most animals really like it. On the other hand, when you get up to a 1 molar sodium chloride solution, now it's the same taste, but it's noxious. It's seawater. And so when you look at this neuron's response, you can see something that I'm going to make easier to see by putting a background on it. You can see that this response, sort of like what we saw in the face cortex data, has 3 phases. There is an initial response that this neuron fired. Not all neurons do this, but there's a burst of action potentials that this neuron fires that basically we call the detection response, because we know that the taste has hit the tongue. And we as experimenters can look at these data and say, yes, a taste has arrived. And so all that that allows us to do, though, is know that a taste has arrived. And then you can see that over the course of the first couple of 100 milliseconds, these responses diverge from one another. And now we're in a situation where just by looking at this period of this response, you can tell what the identity of the taste on the tongue is. We can now code the taste. You'll notice, though, it doesn't look anything like what you would expect given the Zucker data I showed you. First of all, this neuron responds to all of the tastes, just to differing degrees. But we can use any number of simple mathematical techniques to pull out what the taste is. We can identify the neuron. That you can identify the taste that the neurons respond most to and what the taste is from this neuron's response. But then you can see that over time, this response changes again. And by the time you get out past about a second, the response is no longer telling you that just that a taste is on the tongue. It's no longer telling you what the taste is. It's telling you what the palatability of the taste is. And this is important because when you get a taste on your tongue, it is inside your body. There is no passive viewing allowed. You must either make a decision to spit it out or swallow it. And so palatability is the basis of that decision, and this response becomes a palatability response. This neuron responds most strongly out here to the worst taste, a little less strongly to the next worst taste, and it responds the weakest to the best taste. And we can do, again, a simple analysis to show how palatability related this response is. All we have to do is take the known palatabilities of the taste, how much the animal likes to consume each of them, and correlate that with the firing to each taste at each moment in time. And what you see is that even into this identification epoch, that correlation is nearly zero. And then as you move into the palatability epoch, that correlation grows and becomes significant. And by the way, we see neurons that have the opposite response, that fire most to the sweet taste and least to the bitter. But basically, whereas there are all sorts of different cell-specific responses in this identification period, out here there are essentially two types of responses. And so it turns out that what we got wrong here, and I will say this because I just like picking fights with everyone, that when we first saw this stuff, we were super excited because this is a decision the animal's making. The animal has to decide, do I want to consume this or do I want to expel it? And we're seeing a decision variable that is ramping up across time in a way that looks very much like classic integrative decision-making in non-human primates. I think you maybe even had some talks on that yesterday. The idea that there is a biased random walk, the drift diffusion model is what it's usually referred to as, where you have a growth of a decision variable, and when it reaches a threshold, that's when you decide you're going to swallow or spit it out. And so we thought, oh my God, we're seeing what Mike Shadlin and his people have shown in a very different animal, in a very different system. Unfortunately, we were wrong. Because it turns out that when you just look at the data in a different way, it turns out that you don't have this nice gradual ramp into palatability. Instead, what you have is a nonlinear shift in the firing of a bunch of neurons. And so if instead of doing PSDHs, where you're looking at each one of these lines, as I told you at the beginning, is the average of 10 to 15 deliveries of each taste, you instead, in one neuron, you instead look at a whole group of simultaneously recorded neurons and their response to a single trial, you see something very different. And so here are the data. And this is, again, this is actual real data from my postdoc, Lauren Jones. And again, this is neuron 10 fired an action potential there and an action potential there. Neuron 9 fired action potentials there and there and there. And what's in these data, although it's kind of hard to see with the naked eye, so I'll give you a little help, there was a time point right around in the middle of the time that ramp that I showed you with the PSDHs was happening, where suddenly this neuron started firing, this neuron started firing, this neuron started firing, this neuron started firing a bunch more, this neuron started firing a bunch more. At that same time, by the way, this neuron stopped firing, and this neuron stopped firing. And so while we're only recording from 10 neurons, 7 of them suddenly changed their output at this moment in time, which again is in the middle of that period where the ramping of palatability information is supposed to happen. I will, again, in interest of making more fights, I'll tell you that we can see this with 10 neurons. I'm going to show you some more stuff about it. We can see this with 6 neurons, because this is not a sparse thing. This happens. And so one of the reasons I don't use NeuroPixel is because we've gone up to 75 neurons at a time, and we see exactly the same thing. This is a simple process for the animal. The animal is trying to make a decision, and we can see it with few neurons. And so let me show you about how we know that this is the decision that's being made. First of all, by the way, you can't just draw in lines and say that's what happens. We use hidden Markov modeling. 1 second, I'll take your question in just a second. And it basically is designed to show us whether or not the data can be modeled in terms of a pair of stable states with a transition in between them. And so the transition has an uncertainty time. And you can even plot the confidence that you are in this state. And you can see that the confidence is 0, and then it goes quickly up. Yes?
Yeah, these sequences are very cool.
Thanks.
Can you remind us of the brain area? Because it's a crazy low-dimensional population code, right?
Excuse me?
It's a crazy low-dimensional population code.
Yes, and this is gustatory cortex. This is primary taste cortex.
But
I will say,
neocortex.
Huh?
It's in neocortex.
Yep.
Because usually that is considered like more complicated.
Yes, and this is one of the reasons for my skepticism and snark. Is that I think that what we've got, when you hear people talking about how many hundreds, thousands of neurons they need, it's because they don't know what they're looking for. It's because they don't have a good grip on what the process is. We've seen this since, and I'm going to show you in a little while, in every brain area we look at that processes tastes, undergoes this sort of transition. We'll get to that in just a minute. But first I want you to know that, that we do have to explain how we got from that slow ramping of palatability to that sudden transition, how those two live together. And the answer is quite simple. It turns out that on each trial, and this is basically, I've thrown up the hidden Markov model solution to a sequence of 10 trials. And so what I've plotted is this line related to how the system becomes confident that it's in whatever state is dominant there. And what you can see is that on each individual trial, that increase in confidence is precipitous. It happens really fast when you use the population, but you also can see that on each trial it happens at a different time. And if you average across trials like you do for PSDHs, that really precipitous rise disappears. It becomes a slow ramp. And I should mention that we have done, I can point you to the article. I didn't because I figured you had enough to do, but we've done all sorts of analysis on this, and actually it's not just that this rise is fast. To the degree that we can measure, this rise is instantaneous. And so you want to take a break? I'm into the second half. I am in the second half of my lecture, of my whole thing. Take a break and come back? All right, and so we're going to come back and talk more about this after we all drink coffee and pee. Or have lunch. All right, thank you.